Hey, I'm Colin, and my colleagues are Felicity and Rohan. We research on the design and screening of palmatine derivatives as G-quadruplex binding agents. So in our research, we search for the characteristics of palmatine derivatives that lead to relatively high binding affinities to the molecular complex G-quadruplex, also known as GQUAD. The scaffold we chose for our molecules is palmatine, a protoberberine alkaloid known for its anti-cancer properties. We created a molecular library of over 100 ligands based off of the scaffold, and then docked each molecule to GQUAD using AutoDock Vina, an open source software application for modeling simulations. The generated binding affinities were recorded, and analysis was conducted on the entire library to determine the features of palmatine derivatives that bind well to GQUAD. And now I'll pass it on to Rohan for the introduction.
G-quads or G-quadruplexes are molecular complexes formed by the folding of guanine-rich DNA. They are formed by Hoogsteen bonding between guanine base pairs and stabilized by Na+ and K+ ions. They have been frequently investigated as a potential cancer therapy agent due to their ability to inhibit the spread of cancer by preventing the shortening of telomeres, which are the ends of chromosomes. And generally, the shorter the telomeres are, means there is a higher risk of gene instability and cancer. On its own, a G-quadruplex is relatively unstable, so binding ligands are needed to increase its effectivity and stability. Thus, the search for a ligand with a high binding affinity to G-quadruplexes is crucial for the possibility of G-quadruplexes in the future being used as a viable anti-cancer agent in the future. Now I'll pass it on to Felicity for the methodology.
First, we downloaded our Palmatine scaffold from PubMed and imported it to Marvin Sketch, where we created new molecules by adding various functional groups to the base Palmatine. Afterwards, we saved these molecules as CX files so that we could input their code into Avogadro and then optimize their geometries. This then readied them for molecular docking, which we did through AutoDock Vina, as mentioned before, with our chosen G-quadruplex 1KF1. Docking these molecules allowed us to view their binding affinity, which we then put into a spreadsheet along with the aromaticity of the molecules to create a graph as shown.
So here's a scatter plot of the number of aromatic rings by the binding score for each molecule in the library. When creating the library, it was split up into a series of 15 ligands each. Note that the more negative the binding energy is, the better the ligand binds to the complex. Based on this graph, we concluded that there is generally a positive correlation between the number of aromatic rings and binding score, despite the presence of some outliers. On the right, we have the molecule C4GQUAD, which possessed the highest binding affinity out of the others in the series. Here's an image of the binding pose that C4GQUAD takes when the molecule binds to GQUAD, generated using Chimera. And our conclusion?
So far, our group has docked, created, and analyzed for over 160 ligands. Till now, our study has concluded that as the number of aromatic rings increases, there is a general increase in the binding affinity of the ligand to a G-quadruplex. We can see this trend in the graph that is in the results section, as Colin mentioned. Currently, we are trying to research more into how the number of aromatic rings actually affects the G-quadruplex proteins by reading other research papers by other researchers. In the future, we look to create, dock, and analyze ligands with higher binding affinities so we can maybe find something like a specific functional group in a ligand that generally corresponds to a high binding affinity.
Finally, we would like to acknowledge our previous project members for their contributions, Mr. Bra, our principal advisor, and ASDRP for their support on this project and for allowing us to pursue it.
All right, thank you everyone, and that was our presentation.